Open the pod bay doors, Tom.
I'm sorry, Dave. I'm afraid I can't do that.
What would you do with a brain if you had one?
Do? Why, if I had a brain, I could...
Miles per hour.
I could while away
46, 56 degrees.
And I wanted to start by asking you, so you made this film, I think it should be on, but you made this film in 2000, and science, specifically biology, but also technology, both the technology of filmmaking, but also computer technology has advanced a lot. I'm curious about what your thoughts are seeing the film today in 2017.
Well, I'm amazed at how accurate and relevant it is
Yeah.
in these times. I have to say that when I made it, I wanted to also create the portal. And when we made it, we actually achieved the fact that there were Palm Pilots in that time. And when you sat in the audience and had a Palm Pilot, the Ruby program would leap off the screen into your pocket and you'd leave and be able to talk to her.
Oh, wow.
In the
so
old
you did
days.
that in theaters, actually?
Yeah, we did. And then Dina is an extension,
Right.
which is at the Whitney right now of that AI version that began here. But I think what's happening in science is in biology right now is really we're at a crucial point in being able to genetically engineer, and anybody, as you know, genetically change and re-edit living things in a profound and remarkable way.
Well, I have to say what I found remarkable is that the film was made in 2000, is that right? Which predated actually the Human Genome publication by 3 years. So it was quite remarkably advanced then. I mean, I guess the project was going on. I assume you knew about it.
Yes, I did.
But it wasn't complete until 2003, I think, when it was first announced. So, yeah, so I was shocked, actually, when I found that the film was made in 2000.
Yeah.
Now I want a portal, too. I want to get on that portal. What happened to that? Is it only for the Palm Pilot?
It was only for the Palm Pilot.
See, so that we
at
couldn't
the time,
predict, right?
Yeah. Nobody
Saw that, did
right.
we?
The distribution was very limited. The film of the film it's being distributed more now than it was then. In fact.
Yeah, so Stuart, I'm curious, one of the things I think that we can say about something that happens in the film is that it's an example of asexual reproduction in that, Rosetta Stone is creating these 3 replicants. She's herself is not having sex with anyone. I'm curious if you can, or if you know of any plants or animals that do that in nature, and also if asexual reproduction can ever produce males.
Um,
Or wherever you want to go with that.
wherever I want to go with asexual reproduction.
Yeah.
Well, all right. I'll try and stick to the biology here. So there are animals that, well, of course, so very primitive animals, bacteria, what are called prokaryotes. I'm sorry, I don't want to throw a lot of stupid terms around, but early kinds of single-celled animals that don't have a nucleus, which is why they're called prokaryotes. So bacteria, archaebacteria, things like that, certain kinds of protozoa, they only produce asexually, which is they just simply divide and multiply, as it were. Then as soon as you get to real cells, so-called eukaryotes, you begin to see more sexual reproduction. And it's still, I think, a pretty open question in evolutionary theory as to why one exists versus the other. There are advantages to both. If you want to reproduce really quickly, asexually is the best way to do it. I guess that's obvious. If you want to just make a large number, if you want to invade a niche or an ecosystem like a bacteria might, like in an infection, for example, then you would reproduce asexually, just make a large number. The idea of sexual reproduction, we think, there's a lot of hand waving, is that it produces variety. And so it enables a species or any kind of an organism to confront lots of variation in the environment successfully. But there are animals, multi-celled animals that produce asexually, plants, some fungi, and some animals. Some animals do. I guess sometimes it's called parthenogenetic. reproduction, and as far as I know, at least in mammals for sure, you can only get females.
Yeah.
In the rare, or I shouldn't even say, I don't even know that there are any mammals that do it, but in reptiles, fish, any. So I know we all think that most sexual I'm sorry, am I giving a lecture here?
No.
Oh, okay,
I mean,
all right.
it's interesting.
If I start giving a lecture, stop me.
Don't worry.
We all think of sex as being X and Y chromosomes, but there are other forms of sexual reproduction. There are W and Z chromosomes in different animals and things like that. So among X and Y chromosome animals, which includes all mammals and most vertebrates, then I believe you can only get females from asexual reproduction. But among certain insects and other critters like that use this WZ form, which is more complicated than we want to talk about, then I believe you can produce males occasionally.
Huh.
I'd like to know what you think of the CRISPR technology.
Yeah. I guess we have to get to CRISPR. So now, yes, you're right. I mean, you said gene editing earlier, and so there's this new technology available now in biology called CRISPR-Cas9. CRISPR, CRISPR, is an acronym which I can never remember. It's a very complicated acronym, and the Cas9 is a capital C, small a s dash, the number 9, and that refers to a particular protein. So this technology, this CRISPR-Cas9, which you may have seen in the paper or heard talk about or whatever, is a new form, a new mechanism for editing genomes. It's extremely accurate, and although very hard to understand how it works, it's a very complicated mechanism. It's actually trivial to do. I mean, you could do this literally in your kitchen. I mean, it's really a recipe.
Right.
I mean, you really saw that, I have to say. That is scary.
And you could have it on a flash drive, and
Yes.
It's this big, and you can edit living forms. And there's no FDA, there's no rules about who can edit what or what's going to happen globally.
Right. I mean, not yet. I mean, people are talking about it and trying to decide what should be done about it. But, and of course, you don't only have to have rules. You have to have people that are willing to obey rules. So that doesn't always happen. In this case, it would be extremely difficult to enforce rules because it's so easy to do. And so there's a lot of worry about it, but there's also tremendous optimism about it because it will allow us to do things that just were unimaginable or were a kind of a film, not quite fantasy, but you know what I mean, nonfiction kind of, a fictional film. Now, I mean, this is the kind of thing that could almost be done. Building a human being from scratch is a little tricky still, I think, but, well, it's not tricky if you do it sexually, of course.
They're doing it.
It's tricky to do in a bench top somewhere, well, it's tricky to do in a lab, let's put it that way.
Isn't it true that you can grow human cells in a petri dish? Aren't they, do you know?
That's a good question. I mean, you can grow human cells in a petri dish. I'm not sure you can make them. You mean make a human cell?
Do you know why they don't put human cells, that they don't use human cells in these transgenics anymore?
No. In which transgenics?
In putting human life into...
into other creatures.
Because it's a violation of the 13th Amendment, which is slavery.
Really?
Yeah. And that if you own a cell from a human, you own that human. Therefore, so that was the Supreme Court decision because they had been doing it to start with, and then it was stopped here. But they just allowed it to be done in England.
Right. I just saw. I didn't read the paper, but I saw this paper about growing. I mean, there's always this issue of trying to grow human organs in other animals, right? So that's human cells. So I don't know where that fits in with that. How does that fit in with that? I mean, you can do that, right? Or at least people are trying to do it, I think.
I think with bioprinting for organs, it's different than actually putting
a cell in that can replicate and all the rest of that kind of thing. That could be. That could be. I'm actually unaware of that.
So Lynn, I want to ask you. So in the film, Tilda plays these 3 self-replicating automatons, and since we're speaking about genetics and how did that concept emerge? Like, is there anyone for you? Is there anyone who you talked to in particular, or research you did?
Yeah, I read a lot, and obviously at the time, and I'm getting ready to do another one soon. I've been talking to people for the last 3 years about this. In fact, I'm going to CRISPR George Church's lab at Harvard in a couple weeks. So yeah, I mean, but it's in the air. I mean, you don't really have to look far to
Literally
get
in the air,
information
I'm
about
afraid.
this.
Yeah. Yes, no, I mean, it's all out there. I mean, I would say, there's a Nobel Prize afoot in this. There's a huge patent battle going on over it between 3 laboratories at Berkeley and at Harvard and now in Germany at another laboratory there. And there are different players in it as well who are all, it's the whole patent thing and all that. Because it clearly will be worth billions and billions of dollars as a technology. I just like to make a quick pitch here. This is, unrelated to the movie, but I'm just going to make a quick pitch for the fact, well, not entirely unrelated, quick pitch for the fact that the 2 people who made the initial discovery, 2 women, Jennifer Doudna, who's at UC Berkeley, and Emmanuelle Charpentier, who's now at the Max Planck, and George Church and forget his first name, what's his first name? I can't think of his first name. First used it in human cells, or showed it could be used in human cells. But Doudna and Charpentier were slugging away in the trenches for years and years doing what we call sort of fundamental or non-applied research, just curiosity-driven research. And what they were trying to understand was the immune system of bacteria. That may sound pretty archaic, and it's arcane, and it is. But bacteria are attacked by specific viruses that only attack a certain kind of bacteria. They don't get to us or any other critter. They just are interested in bacteria. But bacteria, unlike us, don't have an immune system, so they have no way of protecting them from these viruses. So they use a variety of genetic tricks to chop the virus up, to chop the viral DNA up before it can take over the bacteria's DNA and kill it and use it to replicate. And CRISPR was a technique that bacteria developed for this, and it took over 30 years of really difficult research, not well-funded research, by the way, because nobody could understand what value this would be for anything. It was just an interesting thing to know about. But Doudna and Charpentier discovered this, and then with the discovery of this Cas9 protein, then immediately became obvious that this was a gene editing technique, that you could apply this to gene editing.
You know what I'm really interested too? I did an interview with somebody at Oxford about 6 months ago, and he was talking about epigenetics and how you can alter if trauma travels genetically through your DNA, and they could locate it now and operate on an embryo prior to birth. He said, to correct something before that child is born, so that they won't have to spend a life correcting it with chemicals.
But you mean operating on their genes?
Yes.
Not cutting them open,
right,
or anything like that.
Right.
Yes. No. That's exactly what you could do. You could now find that, assuming you knew the gene that was causing this, which we sometimes do and sometimes don't. But if you know the gene, I mean, a perfect example even would be cystic fibrosis. Cystic fibrosis is caused, 70% of the cases of cystic fibrosis are caused by a single mutation in a single DNA base pair in a molecule that makes a protein that's 2,000 amino acids long. So it's 6,000 base pairs, and one of them is mutated. And that causes 70% of the cases of cystic fibrosis. That could be trivially corrected by using CRISPR in an embryo which had been tested for this gene. And if you saw it had 2 copies of this bad gene, you could correct, all you had to do was correct 1 copy, in fact, and there would be no more cystic fibrosis. So so that's the power. That's the good side of it, right. Right. Now, what other things you could do, as you know?
Well, like everything. I mean.
Yes, it's Golem-like, I mean, there's always the golem in the room, as it were.
Well, so
It's a great line.
so in the ZKM's catalog of your work, Lynn, called Civic Radar, Tilda Swinton wrote you a letter, and she said to you that in your work you never ask what if, only why not. And
very nice.
I think that you're dealing in these realms, like morally ambiguous realms, I think, of science as we're talking about, and I just wonder, would you say anything to her? Do you agree with that? Like, is that how you see your work? Or?
Yeah, I think if you have an idea, you're almost obligated to carry it through and to think of ways that it could be used as a creative force for preservation, in the world. So, and that's part of the fun of it. Is not having the limitations of rules that have been imposed for some other reason that doesn't apply to you or to what's going on right now. So it's really keeping in the present.
I have to say, one of the things that I really enjoyed about this film was, I think in other hands, if you will, it could have been just another one of those dystopian gene editing
It has
gone
been.
wrong
It-
things.
it has
Whereas
been.
in your hands, it was not.
Gattaca.
It's quite an optimistic sort of a happy ending. Really, right? So I think that's great. I'm for that. I like
Well,
that.
you know, it's not the technology. You know, technology is neutral. You know, it's what we what we do with it, an intention.
Yeah, so I'm curious, and other people might be too, in that Tilda is playing these 4 different roles, and do you know if she did any particular preparation for any of them? Did you give her any, specific direction? I'm sure you did, that you can remember.
She was really interested in who was born first. and the personalities that they would have in that way.
Uh-huh.
But in general, I mean, she's so smart, much smarter than I am, and carried things to levels that I couldn't have dreamed of. So, you know, you just get the best people you can and set them loose.
Yeah, and you've worked with her before on
Yes.
many projects.
Yeah.
Yeah, so is this, so I read, I think you referenced this earlier, but that this film is supposed to be the second in a trilogy.
Yeah, I'm trying to do the trilogy
third.
at some points.
So can you say what the third might be? Or
Do I what?
do you know what the third?
Yeah.
You do?
I do.
Could you tell us? Please.
It's about two sisters who operate a lab together, and they create a creature using CRISPR technology that where they use a dolphin cell so that that the child would have increased perception of the sound of the world and could be able to have auditory possibilities of seeing when the planet's in danger. But then the child becomes an adolescent and thinks that
How dolphins are when they're adolescents.
Yeah.
I have somebody for you to talk to, actually.
good.
My wife is sitting in the audience. She works with dolphins.
Oh, great.
Studies their acoustics, in fact.
Yeah, Diana Reese. Everyone should look up her work.
Cool.
Yeah, so
Dolphins and CRISPR, that's really got me going.
yeah, so I mean, given that art has this possibility of, sort of giving us another view onto the culture and the climate that we're living in, do you see your films like making any specific interventions into like how we think as a culture? I mean, does that make sense?
I think that they open up different kinds of possibilities. You know, particularly, certainly the last 3 documentaries, you know, Women Art Revolution, Strange Culture, enabled somebody to be freed from a potential prison sentence. Women Art Revolution inserted previously eradicated history into culture. The film I just finished, that's going to premiere in 2 weeks, you know, is about PTSD and trauma, and how you convert the psychology of that into a positive creative force for reversed thinking. And I think all of these ideas come out in the world, and people take what they can, what take things from them and adapt them in some form, just putting them into the world.
Can I ask her a question?
Yeah, of course.
Okay. So where did this interest in science begin?
Everybody in my family are scientists, but me. I'm the black sheep.
Congratulations.
I'm the black sheep.
No.
My daughter's at Columbia.
Oh, is she really?
Yeah.
Oh, in science?
In science.
Oh, all right, we'll talk about that. Okay.
My mother's a biologist.
Really?
Yeah.
Huh.
So did you ever think you wanted to be a scientist?
I had no choice. I mean, I majored in biology, in fact, but I was always doing art things. So
Well, so this gives you a very interesting kind of perspective on it. Though,
I think
right?
science is magic.
Yeah,
You know,
yeah.
it's the real magic
Yeah,
of our life.
I agree.
Yeah.
Yes. Yeah. Well, it's very nice. It's very nice to see it done this way, though. That's great.
Yeah, and I mean, Stuart, I don't know that most people know, but you came to science later in life. relatively speaking.
After a long prison sentence. No, that's not
In
true.
the theater. So,
Yes, almost.
yeah, I don't know if you want to talk about, you've written about ignorance and failure and sort of the process of investigation that comes with doing scientific research, and I wonder if, having been in a little bit both of these worlds, if you have any thoughts about the similarities or differences in these fields. Yeah.
So what she's referring to is that I did work in the theater for a number of years before going back to school. Actually, before going to school, I never went to undergraduate, at least till I was about 30 years old, because in those days, you wanted to work in the theater, you apprenticed yourself to somebody, you know, and that's how you kind of did it. And so then I went back to school because of this kind of vague interest in animal communication was what I thought I was interested in, but I got deeper and deeper into biology somehow or another. And then wound up giving up the theater and doing this biology business. I now work on the sense of smell. That's what my laboratory works on, as a kind of a window onto brain mechanisms or function. What do I see the same in them? Well, I don't know that there's anything the same in them, certainly not just because they both attracted me. I think different things attract people at different times of their life for all different sorts of reasons. I love science. I mean, I do think that science has many of the things in it. You probably have this feeling too, from your family at least, that the theater and film does as well. I mean, aside from ignorance and failure, I have these two books. I'm kind of carving this little niche out for myself here. But I think ignorance drives science in the same way that it drives art. It's what you don't know that's always most interesting, what problem there is to solve, not the stuff you do know. As soon as you know how to do something well, you're done with it, kind of, right? And then the other thing is this willingness to fail, not in this kind of way of, oh, failure, you know, is good for your character or something like that, but really failing, understanding that failure is embedded in the process. It's woven deeply into the process. And that if there isn't sufficient failure, then nothing will. It doesn't matter. The failure is not good because retrospectively it became successful, or eventually it led to success. It's just good on it. I think Gertrude Stein said, real failure needs no excuse. It's an end in itself. And I think that's correct, I think. You never know with Gertrude Stein, I guess, really, do you? But I think that's correct. So in that sense, I think they're very similar also, that science works mostly with failure, and I think, I'm sure you would say your process is mostly about failure, right?
Totally.
Yeah, there you go. See? So I think
And
in that
then you
sense-
wait and you find out that they're not.
Right. And then finally...
Like this film.
Mm-hmm.
Yeah.
People hated this film
Yeah,
when it
is
came
that
out.
right?
I couldn't get it distributed at all.
Well, then, I'm trying to think back to 2000. I could maybe see that. 2000, yeah.
They said it would
Well-
be a minor footnote in the feminist sci-fi world.
Well,
The New York Times did.
To them. Oh, well, but you know, the New York Times is, you know about them now, so. The truth is out with the New York Times.
Did you get any responses from the scientific community at the time that it was
Oh, that's a good
released?
question.
Nobody liked the film at all.
How about your mother?
me
Did
and
your
Tilda.
mother like the film?
Me and Tilda liked
You
it.
and Tilda, that's a good crowd.
And we giggled our way through it, but and she always shows it at her retrospectives, but
Yeah.
not very many other people. But it's growing. It's growing with your generation, in fact. So
Yeah, why do you think that is?
because they get it.
Yeah, something.
Thank goodness for the millennials.
Yeah, somebody's got to take over soon.
Yeah, I actually thought that one of I love the part in the film where Sandy is conducting in the Xerox Lab, and you once wrote a piece about, for Science and Film actually, about how you first came to work with technology. Like how, you know, your first interactions with technology in your work, and I think you said that it began with a Xerox machine.
Yeah.
I wonder if you could,
I was, maybe 15 years old, and I was trying to reproduce a photograph, and it got stuck in a Xerox machine, and it was so much better than the drawing. I really liked, it was ripped up and crinkled, like the
wavy
image up
line
there.
thing,
Yeah, and I realized, that it was a symbiotic thing, that that maybe the Xerox machine had its own mind and was correcting what I had done.
I have to say I like the orchids, by the way. I thought that was a nice touch. Orchids, have among the weirdest sex habits of any plant.
I didn't
in the
know.
world. Oh, you didn't know that?
No.
The orchids weren't intentional?
Well, tell me. Maybe I'll use it when you're not around and say they
But
were.
please do. Oh, yeah, so orchids have, first of all, they reproduce bee pheromones, and there's several different animals that pollinate different orchids, and they each one produce a scent that's specific to that critter. It's literally the pheromone that animal makes, which is not trivial to figure out how a plant figured out what the pheromone of bees was and then made it. I mean, didn't figure it out, of course, it evolved in some way, but it's not a trivial kind of problem in evolution. And then when there is no pollinator around and there is no wind around, this plant actually can move and, if you will, fuck itself. It's a hermaphrodite. So, all plants are kind of hermaphroditic. They usually have both the male and female organs. And in this case, it'll just asexually, if you will, reproduce. Well, sexually asexual. Hermaphrodites is not asexual reproduction. It's actually sexual reproduction. It's just done by yourself. It's biology. What can I tell you? But anyway, so I thought the orchids were sure there on purpose because they're. Oh, okay.
They were.
She couldn't have lucked into that one.
Yeah, I mean, the other, there's so much to appreciate in the movie. The other thing is that I wanted to ask about are the costumes, because I read in the credits that Yoji Yamamoto, who's a Japanese fashion designer, did the costumes. And so I'm curious about that, and also whose idea it was for them to be color-coded.
Yeah, it was RGB in the colors of video.
Right.
Cool.
But I mean, it's sort of not. I mean, he gave us the clothing, except for when they were doing the dance and doing other things where you had to be in the same room. We had
The
to
green screen. Yeah.
exactly. We needed something to get in and out of quickly, so that's why we used the kimonos.
Oh, so he designed the kimonos?
He didn't.
He didn't. Oh,
But-
it was
but the other
everything else
clothing, yeah.
besides that. Got it. Okay, cool.
Yeah.
Okay, so we have to wrap up, but I want to, again, encourage all of you to come across the street, join us for a drink, continue the conversation. I want to, thank Stuart and Lynn so much for being here.